Hey everyone, Dr. D here, and in this video we are covering Chapter 8 from our Talaro's Foundations in Microbiology 12th edition, covering an introduction to microbial metabolism. Let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
Now, this is one of those chapters that goes into details where they assume that you've already kind of learned certain concepts before, like how enzymes work or how cellular respiration works. So when we get to those parts of the chapter, I'm going to link to other videos I've put together above, I'm going to make a card above my head. And those are going to help you to get up to speed with those concepts. Okay, so we can work through this chapter together. It's probably one of the harder chapters in the book simply because it goes into some of these detail-rich concepts. So let's go ahead and start again, an introduction to microbial metabolism. First of all, what is metabolism? Metabolism includes all of the chemical and physical workings of a cell. So basically, everything that's happening inside of a cell, all the energy that's being used, all the chemical reactions, think of every single chemical reaction in the cell or in your body or in the organism. That's metabolism. And there are two branches of metabolism. There's catabolism and anabolism. Think about these are the two branches of metabolism. Catabolism is degradative, which means it breaks bonds and breaks things down. You're breaking larger molecules into smaller molecules. Think about breaking down fats or breaking down proteins or breaking down sugars or polysaccharides. We're breaking large molecules into small molecules. And when we do that, this typically releases energy. Energy is released during catabolism. Anabolism, on the other hand, involves biosynthesis, the process that forms larger macromolecules from smaller molecules. So here we're building, right? Anabolism involves building. And when you build, this requires energy input. And the two go hand in hand, right? Because the whole reason we consume nutrients, sugars and fats and such, is so that we can break down those sugars and fats via catabolism, and that releases energy, and we use that energy in order to conduct anabolism, to build the proteins, the lipids, and the carbohydrates we need to live. So the two go hand in hand, and remember, catabolism and anabolism together make up your total metabolism. And many of the different processes inside of the cell that make the cell function and allow metabolism to work involve enzymes. Enzymes are usually proteins, not always, but usually proteins, and they are biological catalysts that increase the rate of a chemical reaction by lowering the activation energy. And by the way, now that we're starting to get into enzymes, you can see that these are some concepts that can be a little bit daunting and assume prior knowledge, right? So I'm going to throw up a card at this point. You'll see it at the top corner of your screen right now. And that is a link to my lecture in 1406, Biology 1, covering enzymes in detail and metabolism in detail. So if you don't have the background in metabolism, you could watch my Bio 1 lecture where I go into great detail and you could learn it and get that strong foundation and then come back. So anyway, enzymes speed up reactions by lowering the activation energy, and they are not permanently altered in the reaction. This means that they are not consumed or destroyed during the process. So an enzyme can do the same thing over and over again without getting destroyed, without getting permanently altered. So again, enzymes, here's how they work. if we have the reactants on the left on this graph and the products on the right, then we have the energy state of the reaction on the y-axis and the progress of the reaction on the x-axis. Another way of stating energy state of the reaction is free energy, remember, or G for free energy. Notice how the reactants have a high initial state, a high free energy to begin, a high G value to begin. They have a low G value when they're products. And notice this speed bump. You see this speed bump between the two? How you go from the initial state to the products, but there's a big speed bump. That speed bump represents the activation energy, or here they call it the energy of activation in the absence of an enzyme. And the bigger the activation energy is, the bigger this speed bump is, the slower the reaction occurs. And when you add an enzyme, if you add an enzyme, what it essentially does is it lowers that speed bump. It lowers the activation energy. And the lower the activation energy goes, the faster the reaction proceeds. In fact, if you were able to lower this speed bump all the way down to here, then it would be an instantaneous reaction. And so again, that's what enzymes do. Enzymes are usually protein catalysts. Catalysts speed up a reaction, and these enzymes do not get consumed by the reaction. They can do it over and over and over again, and the way they function is by lowering the activation energy. And when we're discussing enzyme structure, there are simple enzymes which consist of a protein by itself, but then there are also conjugated enzymes or holoenzymes, which contain a protein and a non-protein component. The protein component of a holoenzyme is called the Apoenzyme or Apoenzyme, which is, again, the protein portion of the enzyme. And then there's also a cofactor involved with holoenzymes. The cofactors are not made of protein. Instead, the cofactors are either some sort of inorganic element, such as a metal ion like iron, copper, magnesium, something like that, zinc, or an organic molecule such as vitamins. Do you guys remember when I said vitamins are organic cofactors and minerals are inorganic cofactors? That's exactly what I'm talking about. And by the way, the organic cofactors are also called coenzymes. So here you can see holoenzymes. In purple, you have the apoenzyme, and then you also have the cofactors attached here. This is a metallic cofactor, so it's a mineral. Here is a coenzyme, so it's probably a vitamin. Here is a coenzyme, it's probably a vitamin. And then here's a metallic cofactor. It's some sort of element. And these are all enzymes. Again, that's why it's so important in your diet, not just for you and me, but for all creatures, that their diet includes vitamins and minerals, right? You have to have your vitamins and minerals. Why? Because those vitamins are coenzymes that stick to important apoenzymes in your cells, and they help the apoenzyme to work. That forms the holoenzyme, and only then does the enzyme function correctly. And the minerals are the same way. The minerals can attach to apoenzyme and then allow the holoenzyme to function correctly. So if your diet lacks particular vitamins like this or minerals like this, well, then that enzyme's not doing its job correctly in your cell, and that could have an adverse effect on your health. And here is a nice table showing you different enzymes on the left and the cofactors they require on the right. Obviously, you do not need to memorize this whole table, but notice how some enzymes like catalase require cofactor that's inorganic, like iron, while others, like succinate dehydrogenase, require organic cofactors like FAD. And then yet another one, pyruvate dehydrogenase, requires both an organic cofactor, thiamine, and an inorganic cofactor, magnesium. Isn't that neat? So again, these vitamins and minerals are required as cofactors for apoenzymes to form the holoenzyme so that it functions correctly. And do you recall from previous classes how proteins fold? Proteins start out as a chain of amino acids, which then fold to form secondary structures like alpha helices and beta sheets. And then that finally folds in on itself to form what's known as the tertiary protein organization. This is the final folded protein. And that folded protein, each protein folds in a particular way so that it has a very special site. If it's an enzyme, it's going to have a special site called an active site. This is the catalytic site. This is a site that's folded perfectly to accommodate the substrate that the enzyme is going to act upon, right? So the substrate binds to the active site on the enzyme, right? Why? Because of the perfect folding of that tertiary structure of the protein. So here you can see exactly what I'm talking about. A protein starts out as a long chain of amino acids, which then folds into alpha helices and beta sheets, which are spiral shapes and flat areas, respectively. And then that folds in on itself to form the final folded protein called the tertiary structure. And notice that the final folded protein has a site called the active site. And it's a perfectly folded three-dimensional site which accommodates whatever the enzyme breaks down. So whatever the enzyme breaks down fits into this active site, and then it gets broken down, and then products are released. Okay. And if you have a quaternary structure, by the way, a quaternary structure is when two or more proteins work together as a unit. And again, I know all of this is very kind of in-depth with a lot of details, but please watch that video I linked a little bit earlier on enzymes, and you'll get this whole thing. It's a very in-depth lecture about how enzymes work, what protein folding is, what's the tertiary structure, what are cofactors, what is the activation energy. All of that is explained in that video I linked just a little bit ago. And again, what do enzymes do? Enzymes are these perfectly folded structures, right, of protein, and they have active sites right here. The active site fits the substrate. The substrate is the substance that the enzyme acts upon or breaks down. The substrate fits like a lock and key into the active site of the enzyme. Any other molecule does not fit. Other molecules do not fit in the active site because the active site is specific for a particular substrate. When the substrate fits into the active site, then remember what the enzyme does. It lowers the activation energy, which allows the substrate to be broken down. Usually that's what happens. The substrate gets broken down and then products are released just like that. And again, like I mentioned, some cofactors are inorganic. These are the metallic cofactors, or minerals they're called. These activate enzymes. And then there are the coenzymes. Remember, these are the organic cofactors. And vitamins are the most common type of coenzyme, or organic cofactor. Now, some enzymes are actually secreted outside of the cell. Those are called exoenzymes. And some enzymes work inside of the cell. Those are called endoenzymes because that's what those prefixes mean. Exo means outside, endo means inside. So exoenzymes are transported extracellularly. That means they're released from the cell, where they break down large molecules, or harmful chemicals. Examples are cellulase, amylase, penicillinase. All of these are exoenzymes, enzymes that are secreted by the cell to the outside, and they function outside of the cell. Endoenzymes are retained intracellularly. This means that they are on the inside of the cell, in the cytoplasm usually, and they function there. So for instance, most enzymes are actually endoenzymes. And here you can see what I'm talking about. Exoenzymes function outside of the cell. They're produced inside of the cell, but then they're secreted outside, and then they act outside of the cell. Whereas endoenzymes are produced and remain inside of the cell. Next, let's talk about how the cell can build larger molecules. That's called synthesis. Or break down large molecules. That's called hydrolysis. Let's talk about that next. So when the cell wants to build a macromolecule, then it needs to do synthesis or synthesis reactions. This is known as a condensation or dehydration reaction. In this reaction, one water molecule is released per bond made. Here's how it works. Look. Here is an enzyme that's about to link two glucose molecules to one another. Here's a glucose, and there's a glucose right next to it. These are two separate monosaccharides of glucose. And what this enzyme does is it's going to link these two sugars together. And the way it does that is by removing water. And do you guys remember what water is? What's the molecule of water called? Can you beat Wicket? That's right, Wicket. H2O, right? H2, look, H2, two H's and an O. So do you know what this enzyme does? It does a process known as dehydration synthesis or dehydration reaction. It removes water in the form of this hydroxyl group, this OH, and this H over here. So by removing this hydroxyl group and this hydrogen, removing that as what? As water, now you have formed a bond. It's called the glycosidic bond. You have formed a bond between these two glucose molecules, and you formed what's called a disaccharide. Now the two glucose molecules are linked together as a disaccharide called maltose. Isn't that neat? So that's how your cells build maltose, the disaccharide. It's by removing a water molecule, by removing these two H's and that O. Isn't that neat? And that's called what? That's called condensation reaction. It's called dehydration reaction. It could be called dehydration synthesis. All of those mean the same thing. Isn't that neat? Conversely, how do cells break things down? Cells break down macromolecules by a process known as hydrolysis. So let me show you. Hydrolysis is actually the converse reaction. That means the opposite reaction to condensation. Here, we're using water, one water molecule to break each bond. So watch this. Here we have two amino acids. These two amino acids are linked together already. You see that? These amino acids are linked to one another via a bond called a peptide bond. And I want to break that bond. I want to break that because this is a type of catabolism. I want to break that bond. So how do I break it? Here I have to introduce water. See, you have to introduce water to break bonds or remove water to make bonds. Isn't that neat? When you introduce water to break bonds, that's called hydrolysis because hydro means water and lysis means break because you're using water to break a bond. And if you want to make a bond, it's called condensation or dehydration reaction because you're removing water. Does that make sense? To make a bond. Remove water to make a bond, add water to break a bond. That's what these enzymes are doing. Isn't that neat? So look, it's using water to break that bond. You now have a broken bond. Here is the O, the O and one of the H's, and here's the other H. Isn't that neat? And that now the enzyme releases the two separate amino acids. So again, many of the anabolic processes inside of your cells occurs by dehydration reaction or condensation, by removing a water molecule to build larger molecules, by making bonds. Whereas catabolism occurs in your cells by hydrolysis. Enzymes use water molecules to break down larger molecules into smaller molecules. Isn't that neat? So that leads us to our first concept check. Which of the following statements about enzymes is correct? A, they show no substrate specificity. No, remember, that's anything but true. Remember, that active site is folded so perfectly in that tertiary structure of the protein that it perfectly accommodates a particular substrate. B, they are consumed by the reaction. No, remember, enzymes catalyze reactions without getting consumed by the reaction. C, they may be active extracellularly. That's true. Remember, there's a whole class of enzymes called exoenzymes, and they're designed to work outside of the cell. That's what extracellularly means. So it should be C. Let's see why D is wrong. They are composed only of protein. No, most enzymes are made up of protein. Some include RNA. Those are called ribozymes. And remember, the holoenzyme includes an apoenzyme plus some cofactor as well. So the best answer is actually C, though D is a tricky one. And remember that enzymes are proteins usually that fold in a particular way. These enzymes can become misfolded under certain conditions, specifically these environmental conditions like extremes in temperature, like extremely high temperature, I should say, differences in pH, let's say acidic environments or basic environments, osmotic pressure. All of these things can cause a protein to misfold, and that's called denaturation. When a protein or enzyme denatures, that means it has misfolded due to some kind of unfavorable condition. So because of this, proteins and enzymes are known as labile, or chemically unstable enzymes. And a lot of times during metabolic pathways with anabolism or catabolism, you see not just one enzyme doing one step, but you'll see multiple enzymes doing a stepwise, highly regulated process. Now, there are also substances that can prevent enzymes from working. These are called inhibitors. And there are two main classes of inhibitors. There are competitive inhibitors and non-competitive inhibitors. Competitive inhibitors stop enzymes from working by binding to the normal active site and preventing that active site from binding to the true substrate. In non-competitive inhibition, the substance, the inhibitor, binds to some site other than the active site, but it still has the same effect. It turns off that enzyme, or it makes it so that the substrate cannot bind to the active site. So in this figure, you can see how a competitive inhibitor would work. You see in purple, you have your enzyme. The enzyme would like to bind to its normal substrate in its active site in order to lower the activation energy and break it down into products. However, the competitive inhibitor usually has a very similar shape to that normal substrate, so it will fit into the active site instead. And, if the substrate binds, that's great because you can proceed with the reaction. However, if the competitive inhibitor takes up that active site, well then the reaction is blocked. The enzyme cannot do its function. And do you remember from the nutrition chapter that we talked about how organisms require hydrogens, or they require electrons? Do you remember that? And I said that the reason organisms require electrons is for the electron transport chain, right? To fuel the electron transport chain. And there's an electron transport chain during photosynthesis. There's an electron transport chain during cellular respiration. And so we need electrons in order to make those processes work. And the process of taking electrons from one substance and handing those electrons to another substance are known as redox reactions. Do you remember the acronym Leo says GER? That's a good one to remember if you want to remember what the terms oxidation and reduction mean. Oxidation refers to loss of electron, right? So loss of an electron is oxidation. That's the Leo part of Leo says GER. And then gaining an electron means reduction. That's the GER part of Leo says GER. So just remember that if a substance loses an electron, then it's become oxidized. If it gains an electron, then it's said to be reduced. So take a look at this little equation at the bottom here. You can see that glucose during cellular respiration, glucose is losing electrons. See, because each one of these 12 hydrogens has electrons. Those electrons are getting plucked away, right, to CO2. And the electrons end up on oxygen. So glucose becomes oxidized to CO2, while oxygen, during cellular respiration, did you know that oxygen is the terminal electron acceptor? Oxygen picks up the electrons at the end of the electron transport chain. So oxygen gains electrons and becomes reduced, right? GER, gaining electrons means reduced. Oxygen becomes reduced to water during cellular respiration. So you see how oxidation and reduction work. Oxidized means losing an electron, and reduced means gaining. And do you remember how this process works in cellular respiration? Electron carriers are required for carrying those electrons from the sugar to the electron transport chain. And one of those electron carriers is called NAD+. NAD+ is short for nicotinamide adenosine dinucleotide, and this is known as an electron carrier. What this carrier does is that the oxidized form of the carrier shows up to pick up electrons, 2 electrons, and a proton, and becomes reduced to NADH. NAD plus picks up electrons. That means, remember, if you pick up electrons, you gain electrons, you become what? That's right, Wicket. You become reduced. Now, this is part of cellular respiration. Do you guys remember cellular respiration? Because that's the next topic that's coming up in the next couple slides here. I don't have time to completely explain cellular respiration for this chapter, but I have a video right here above me. There's a card. If you don't remember cellular respiration, or if you've never had cellular respiration, I know some of you may have skipped BIOL 1, but that's where cellular respiration was explained during BIOL 1. And that video I linked just a second ago above my head is my best attempt to explain cellular respiration so that it makes perfect sense. So I really recommend watching that video to get an understanding of cellular respiration, especially if the next few slides, are confusing to you. So let's solve this concept check. And then we will take a short break time with Gizmo and Wicket. What do you say? So concept check number 3, if a molecule has been reduced, right? Remember GER, reduced during a reaction, it has lost oxygen, no. Gained oxygen, no. Lost electrons, no, that's oxidized. Gained electrons, that should be the answer is D. Remember, because GER, gain of electrons means reduction, right? So there you go. All right, welcome back from break time with Gizmo and Wicket. So carrying on, remember the whole point of cellular respiration is to make ATP. ATP is the metabolic currency. It's the energy currency of a cell. It's actually made up of three parts. Adenine, which is a nitrogenous base, that's the same adenine that you find in RNA. You know how RNA has A's, G's, C's, and U's? This is the same adenine as RNA. And you have the ribose group right here, a ribose 5-carbon sugar. And you have three of those phosphate groups. That's where the TP comes in. Triphosphate, right? 3 phosphate groups. And it's that removal of the terminal phosphate that releases energy. So you can see here, here's ATP. Here is your adenosine nucleotide in orange, your sugar ribose, the 5-carbon sugar in red, and then here are your 3 phosphate groups right here. And by cleaving or hydrolyzing, remember we talked about hydrolysis, using water to break a bond. So imagine using water to break this bond right here. That would cause this phosphate group to release, and that releases a bunch of energy that the cell can use to do work. What kind of work do you say? Chemical work, transport work, mechanical work, all types of work in the cell can be accomplished by ATP being broken down into ADP, right? Adenosine diphosphate. And what did you do? You released a phosphate group as inorganic phosphate. And do you remember the steps to cellular respiration? We had glycolysis, which was followed by oxidation of pyruvate. Next, Krebs cycle. And then finally, you had the what's called oxidative phosphorylation, which includes the electron transport chain and ATP synthase. Now, 3 different mechanisms of making ATP are important to understand. And again, I explain all of these in that video that I had linked above earlier, where I linked to my cellular respiration video. I highly recommend you watch that because I go into a ton more detail about cellular respiration. But we need to understand we need to understand the difference between aerobic respiration, anaerobic respiration, and fermentation. And again, all 3 of these are explained in that video that I linked earlier. In aerobic respiration, oxygen is the terminal electron acceptor. You may want to write that down. Oxygen is the terminal electron acceptor. And not only that, during aerobic respiration, glycolysis occurs, the Krebs cycle occurs, and the respiratory chain, which is essentially oxidative phosphorylation. During anaerobic respiration, glycolysis occurs, the Krebs cycle occurs, and again, oxidative phosphorylation or the respiratory chain occur as well. However, the big difference between the two, you should write this down. The big difference between the two is that for the most part, they're highly identical, except in aerobic respiration, the terminal electron acceptor at the end of the respiratory chain or the oxidative phosphorylation is oxygen. Whereas in anaerobic respiration, the terminal electron acceptor is a molecule other than oxygen, like sulfate or CO2 or something like that. Those other molecules are what pick up the electrons at the end of the electron transport chain during the respiratory chain or the oxidative phosphorylation step. And then lastly, what's fermentation? Fermentation occurs when terminal electron acceptor is not available. So for instance, if oxygen is not available, that's when the cell switches to fermentation, which involves only glycolysis, not Krebs cycle, not the respiratory chain, which is also known as oxidative phosphorylation, only glycolysis. And during fermentation, organic compounds are the terminal electron acceptors. So here you can see the three juxtaposed. You have aerobic respiration, anaerobic respiration, and fermentation. Notice how aerobic respiration and anaerobic respiration are highly similar. The big difference is, even though glycolysis occurs in both, oxidation of pyruvate occurs in both, the Krebs cycle occurs in both, oxidative phosphorylation or the respiratory chain occurs in both. But remember, the only difference, you should know, the only difference is what? Can you be a Wicket? That's right, Wicket. The terminal electron acceptor. Here, it's oxygen. During aerobic respiration, oxygen picks up the electrons at the end of the electron transport chain. In anaerobic respiration, one of these other molecules like sulfate, nitrate, or carbonate, they're the ones that pick up the electrons at the end of the electron transport chain. Also, take a look at this. During aerobic respiration, there's a theoretical ATP max of 38 ATP. Some textbooks would say 32 ATP, but this textbook goes with 38. And anaerobic respiration has less than 38. It's never as efficient as aerobic respiration. And then do you remember the last one was fermentation? And during fermentation, glucose is being broken down by only glycolysis. And that means that an organic molecule is the final electron acceptor, such as pyruvate or aldehyde, and you have a maximum of only 2 ATP. So you see how much less efficient fermentation is than aerobic respiration or anaerobic respiration. It only yields 2 ATP because it only goes through glycolysis, not Krebs cycle, not all the other steps of cellular respiration. So for this course, at least know the 3 parts to cellular respiration. You have glycolysis, Krebs cycle or TCA cycle, sometimes even called the citric acid cycle, and then what's called oxidative phosphorylation in most textbooks, but here it's called the electron transport chain plus ATP synthase, right? So I wish they could stick with one nomenclature, but they keep switching up the name of it here. So, and what you should know is what happens during each, as stated on this slide. So look, during glycolysis, glucose is oxidized. That means we take its H's away. We take its electrons away. And so we give those electrons to, remember, the electron carrier. So 2 NADH are generated. NADH is generated because those H's are transferred from glucose to NAD+, reducing it to NADH. Okay, and again, please, if any of this sounds foreign to you, please watch my video that I linked earlier, and I'll put a link down below on that video as well on cellular respiration, because it's too much dense material to clearly explain in this video for micro. So again, during glycolysis, 2 NADH are produced, and when that happens, the glucose gets cut in half into 2 molecules of pyruvate as well, and 2 ATP are produced as well through a process known as substrate-level phosphorylation. Then during the Krebs cycle, again, or TCA cycle or citric acid cycle, here is where more oxidation occurs of the sugar. That means that electrons are transferred to NADH to form NADH, and FADH2 as well are generated, and this causes the sugar to break down to CO2. CO2 is produced during the Krebs cycle. Okay, and then at the end of the Krebs cycle, you also produce 2 ATP by substrate-level phosphorylation. And then lastly, during the electron transport chain, remember, which is part of oxidative phosphorylation, here it accepts those electrons from NADH and FADH2 from the previous steps, and it uses either oxygen as the terminal electron acceptor or a molecule other than oxygen if it's anaerobic respiration in order to produce ATP. And that number of ATP varies, but usually it's 28 ATP from most textbooks. However, I believe in this textbook, it says it's 36. Again, look at this. During aerobic respiration, glycolysis, Krebs cycle, oxidative phosphorylation are all occurring. Oxygen is the final electron acceptor or terminal electron acceptor. During glycolysis, remember, 2 molecules of pyruvic acid are made, 2 ATP by substrate-level phosphorylation, and 2 NADH are formed as well. And then normally, that pyruvate, or also known as pyruvic acid, heads off to the Krebs cycle. However, it can also be used to make various organic molecules for the cell. And it can even be used during fermentation as well as one of those organic electron acceptors during fermentation as well. So pyruvic acid, aka pyruvate, has many different jobs in the cell. It can either proceed with cellular respiration, it can be used to make biomolecules, or it can be used as an electron acceptor during fermentation. So remember, during the Krebs cycle, NADH is formed. See, NADHs are formed. FADH2 is formed. ATP is formed by substrate-level phosphorylation. CO2 is released. And then remember the final step to cellular respiration, you've got oxidative phosphorylation, which includes electron transport chain plus ATP synthase. Here is where those NADH, those 10 NADH from the previous steps come to visit, drop off their electrons in the electron transport chain here, which is colored blue, purple, green, and red. And FADH2, the 2 FADH2 from the previous steps as well shows up and drops off its electrons here as well at complex 2. Those electrons go through the electron transport chain, resulting in active transport of protons. Those protons diffuse back in through a process known as chemiosmosis, exerting a proton motive force, which spins ATP synthase to produce ATP. And remember, the lion's share of ATP is made this way, a process known as oxidative phosphorylation, not substrate-level phosphorylation. So as I mentioned in that overview video of cellular respiration, the whole point of cellular respiration was to get those hydrogens, you see these hydrogens right here in orange, to get these hydrogens away from glucose. And so we formed 10 NADH during the processes of glycolysis, oxidation of pyruvate, and the Krebs cycle. We formed 10 NADH, and those 10 NADH drop off their electrons, resulting in protons being pumped. And during Krebs cycle, we produce 2 FADH2s, and those come over here to drop off their electrons. And those electrons also end up powering the electron transport chain. When you power the electron transport chain with these electrons from glucose, the electron transport chain is then able to actively pump protons into this space here. Remember, this space between the inner membrane of the mitochondria and the outer membrane of the mitochondria is called the intermembrane space. That high proton concentration builds up, and then it wants to diffuse back into the matrix, right? The matrix is in the center of the mitochondria. The protons then diffuse through ATP synthase into the matrix, and that's what powers the ATP synthase to make ATP by joining ADP and inorganic phosphate to form ATP through a process known as oxidative phosphorylation. So that's the overview picture. And remember, a lion's share of the ATP is made this way by oxidative phosphorylation. Now let me show you something cool. This is the difference between a bacterial cell or prokaryotic cell versus the eukaryotic cell. Remember, eukaryotic cells oxidative phosphorylation and several of the steps of cellular respiration occur in the mitochondria, right? Which means protons are pumped from the matrix of the mitochondria to the intermembrane space of the mitochondria. However, this is microbiology, right? So we want to learn about the bacterial cell. Remember, the bacterial cell has the nucleoid, it has cytoplasm, it has a plasma membrane. It also has a cell wall in dark brown. If we take a look at that cell wall, you can see something interesting. Bacteria still have an electron transport chain. See, they still have the electron transport chain. However, the protons get pumped from the cytoplasm inside of the cell to the periplasmic space between the membrane and the cell wall. Isn't that neat? So when NADH drops off its electrons, or when FADH2 drops off its electrons, those electrons move through the electron transport chain. Oxygen picks up the electrons at the end to form water. However, in this case, the protons are being pumped out of the cell into the periplasmic space, where they build up in concentration, and then they are able to chemiosmose or diffuse back through ATP synthase, causing ATP synthase to spin and produce ATP by joining ADP and inorganic phosphate through a process known as oxidative phosphorylation. So isn't that neat that bacteria do the same thing that the mitochondria do? In fact, that's another part of the endosymbiont theory, the theory that states that bacteria were the origin, right, of the mitochondria that were once phagocytosed by an early eukaryotic cell. Isn't that neat? So you can see here that you have the same electron transport chain in the bacteria with complex 1, complex 2, complex 3, and complex 4, as well as ATP synthase. Those are identical or nearly identical between bacteria and mitochondria. However, in this case, the protons are literally being pumped out of the cell. Isn't that neat? So here we can see the theoretical yield for aerobic respiration. That means during aerobic respiration, where glucose is broken down to make ATP, how many ATP are made? Well, it breaks it down here for you. And again, this is different depending on which textbook you're reading, because a lot of textbooks state that the theoretical yield is 32. However, this textbook's saying that the theoretical yield is 38, which is a somewhat outdated concept. So, but we'll go with what this textbook, Talaros, is talking about. During glycolysis, 2 ATPs are made by substrate-level phosphorylation. Then, during Krebs cycle, 2 ATP are made by substrate-level phosphorylation as well. And then finally, during the electron transport chain and oxidative phosphorylation, remember by ATP synthase, 34 ATP are made there. So that should total to 38 ATP total. All right. And that leads to concept check number 5. Which part of aerobic respiration releases CO2? A, glycolysis. No, because remember, I wanted you to know that the products of glycolysis were what? Remember, 2 NADH, and you don't need to know exact numbers. NADH, what else? ATP by substrate level phosphorylation, and 2 pyruvates, right? Or pyruvic acids, as the textbook states. So those are the 3 products of glycolysis. Krebs cycle, yes. Remember, Krebs cycle did produce CO2 and NADH and FADH2 and ATP by substrate level phosphorylation. So Krebs cycle is the answer. C would not be the answer because the electron transport system, all it really does is transport electrons and pump protons, though it does form water because oxygen picks up the electrons at the end of the electron transport chain, and oxygen becomes reduced to water at the end of the electron transport chain. And then finally, D, chemiosmosis. No, chemiosmosis results in ATP production via oxidative phosphorylation, right? So the answer is B, the Krebs cycle. Next, we're touching on anaerobic respiration again. Remember, anaerobic respiration is very similar to aerobic respiration, except the fact that instead of oxygen picking up the electrons at the end of the electron transport chain, from complex 4 of the electron transport chain during oxidative phosphorylation. And by the way, remember when oxygen picks up those electrons, it becomes water. Well, instead of oxygen picking up the electrons, it's some other molecule, some other electron-greedy molecule like nitrate or sulfate or CO2. These types of molecules come along and pick up the electrons at the end of that ride. So for instance, nitrate can come along, pick up the electrons from the electron transport chain and become reduced to nitrite. So NO3 becomes reduced to NO2. So again, anaerobic respiration largely mimics aerobic respiration. You still have glycolysis, you still have the Krebs cycle, you still have electron transport chain with ATP synthase and chemiosmosis. The only big difference is a non-oxygen electron acceptor, something other than oxygen picks up the electrons at the end. And there are many microorganisms, speaking of microbiology, there are many microorganisms that can use something other than oxygen as a terminal electron acceptor. And these are known as anaerobic bacteria. Like I said before, do you remember when I said that not all bacteria grow in oxygen? In fact, some bacteria get destroyed by oxygen due to the ROS, right? Reactive oxygen species that are produced when a bacteria grow in oxygen. And remember from the previous chapters that unless an organism has enzymes such as superoxide dismutase or catalase, they are unable to mitigate the deleterious side effects of ROS. But so these organisms grow where there is no oxygen. You see, they're growing in the absence of oxygen. So if you're growing in the absence of oxygen, that means you're probably breathing something other than oxygen. You and I, we breathe oxygen because we are aerobes. We use oxygen as our terminal electron acceptor so that it can pick up those electrons at the end of the electron transport chain, and that oxygen gets reduced to water, right? However, anaerobic respirators, these anaerobic bacteria, they don't breathe oxygen like you and me. They breathe nitrate or sulfate or carbonate or CO2 or something like that. Something that comes along and picks up the electrons at the end of the ride. And that means they literally breathe something different than you and me. Isn't that interesting to think about? And lastly, remember with cellular respiration, that comes to an end when you low on terminal electron acceptor. So for instance, if I hit the gym too hard and I don't have enough oxygen, that means that I don't have an ability to remove electrons from the electron transport chain. Remember, and everything shuts down except what? Can you beat Wicket? That's right, Wicket. As always, glycolysis, right? Glycolysis is the only process that proceeds. Every other part, the Krebs cycle shuts down, oxidative phosphorylation, electron transport chain, all that shut down. And glycolysis is the only process that's still kind of chugging along, right? So in this case, you only produce 2 ATP during fermentation. And that's kind of a bummer, but that's better than 0 ATP. Does that make sense? And so organisms have to resort to fermentation. And that's because they run low on their terminal electron acceptor. So on the exam, if I ask you the difference between anaerobic respiration and fermentation, you should know the difference. They are not the same thing. Anaerobic respiration involves glycolysis, Krebs cycle, oxidative phosphorylation, the electron transport chain, all that good stuff, except the terminal electron acceptor is something other than oxygen. However, in fermentation, this happens when you run out of your terminal electron acceptor. So you and I run out of oxygen, or yeast run out of oxygen. What happens? They switch to fermentation, right? So you go from cellular respiration to fermentation, and that means that you're only doing glycolysis, not Krebs cycle or any of the other steps, and that's why you only yield 2 ATP by substrate level phosphorylation. And as a consequence of switching to fermentation, you also produce what are called fermentation products. You form acids, gases, other products, depending on which bacteria or which fungus you're talking about. You and I produce lactic acid as a product of fermentation. However, yeast produce ethyl alcohol as a product of fermentation. And by the way, different microorganisms have different fermentation products. Those aren't the only two fermentation products. Again, if you take a look at fermentation here, during fermentation, only glycolysis is occurring. And if you notice, the fermentation products include lactic acid, ethanol, and other alcohols, other gases, other types of substances as well. So here we can see how yeast produce alcohol. Again, for a real step-by-step on alcohol fermentation, please watch that video I linked above earlier, and I'll link in the description below. But this is how alcohol fermentation occurs. During glycolysis, glucose is broken down into pyruvic acid. Remember pyruvate? That pyruvic acid cannot proceed to the Krebs cycle, so instead it gets converted to acetaldehyde, and then finally it becomes converted to ethanol or ethyl alcohol. And that's why yeast produce alcohol. But remember, the only way to make yeast produce alcohol is to deprive them of oxygen, their terminal electron acceptor, by plugging the bottle, right? Plugging that the cork, putting the cork into the barrel so that oxygen is excluded, right? That's the only way that ethyl alcohol or ethanol is made. Isn't that interesting? However, in you and me, right, in humans, that pyruvic acid or pyruvate is directly reduced to lactic acid. See, you and I produce lactic acid from pyruvate, whereas yeast produce alcohol from pyruvate. Isn't that neat? So basically, different organisms on the planet, when they switch to fermentation because they ran out of oxygen, different organisms do different things with their pyruvate. So you and I convert our pyruvate to lactic acid. Yeast convert pyruvate to ethyl alcohol or ethanol. But that's not the only option. Check this out. Some organisms, like Proteus, can produce formic acid and CO2. Streptococcus and lactobacillus can form lactic acid. Acidobacterium produces acetic acid. Enterobacter produces 2,3-butanediol. Propionibacterium produces propionic acid. Clostridium produces butyric acid. And again, yeasts produce ethanol. Isn't that neat? These are all known as fermentation products. So different organisms produce different fermentation products during the process of fermentation. Again, what do you and I produce during fermentation? Can you beat Wicket? That's right, Wicket. Lactic acid. Our bodies produce lactic acid when we run out of oxygen and our bodies switch to fermentation. But look how different creatures on Earth, as shown on this slide, different creatures produce different fermentation products. And us humans take advantage of that. You know, if we want acidic acid, we can use these bacterium and starve them of oxygen. If we want formic acid, we can use Proteus and starve them of oxygen. If we want ethanol, that's what we do when you make wine or beer or alcohol. That's exactly what we do. We put the grape juice in with the yeast, and then we put the plug in the barrel, and we wait until alcohol forms. Isn't that neat? So humans bottle up these fermentation products and use these organisms to make different products for us during fermentation. Isn't that interesting? Again, I know this chapter in particular is very difficult to follow, and so I highly recommend watching my video on cellular respiration that I linked earlier, and I link in the description below, just so you can catch up, you know, because this is really complex material, and you know, they do kind of assume that you've already had some of this material in the past, whether you have or hadn't. So, if you're following along, that's great. If you're struggling, I highly recommend you watching that video I linked earlier. Same thing with the slides that are to follow. Check this out. Next, we are going to talk about photosynthesis, and that's another one of these doozy topics that's just about as complex as cellular respiration. Lucky for you, I have another video, which I'll link above. You could watch it. And that's a complete breakdown of photosynthesis for beginners, right? So that was my Bio 1 photosynthesis lecture. If you need to refresh your mind on photosynthesis, that way I don't have to do a complete new lecture on photosynthesis, as this video is already over an hour long. So during photosynthesis, this is a super important process on the planet. This is responsible for oxygen on the planet. In aquatic ecosystems, 80 to 90% of all photosynthesis is done by these little algae, the cyanobacteria, green sulfur bacteria, purple sulfur bacteria, and even purple non-sulfur bacteria. And that means that they're producing oxygen. They're producing oxygen for us to breathe. Not only does photosynthesis produce the oxygen we need to breathe on this planet, but it also produces the sugars that we need in order to feed off of. Remember, these photosynthesizers are producers. They are at the bottom of the food chain. That means they make up plankton and such. Every one of us consumers feed off of the sugars and the other biomolecules that are produced by photosynthesizers. So if it wasn't for photosynthesizers, all other life couldn't exist, because there would be nothing to eat. And there would be nothing to breathe for us aerobes. There would be nothing to breathe for aerobic respirators. And CO2 would build up quite fast, because CO2 would not be being removed from the environment through carbon fixation. Remember that term from a previous chapter. Now, photosynthesis occurs in only two stages: the light-dependent stage and the light-independent reactions, also known as the Calvin cycle. Remember, during the light-dependent reactions, water is split. Water is oxidized to oxygen, and that's why oxygen gas is released for us to enjoy and breathe. And also ATP is made during the light-dependent reactions through a process known as photophosphorylation, and NADPH is formed as well, right? So in fact, the H's, do you know how H2O has two H's? Those H's, those electrons are transferred from water to the electron carrier NADP+, reducing it to NADPH. So you can think of this H, as the H that originated on the water that was split during photolysis, releasing oxygen. Okay, and then do you remember the ATP? The ATP from the light-dependent reactions, as well as the NADPH from the light-dependent reactions, as well as CO2 from the atmosphere, come together during the Calvin cycle in order to produce what? Can you beat Wicket? That's right, Wicket. Glucose. The whole point of photosynthesis is to make this glucose, because that's what's going to allow you to feed yourself. So for my purposes, you should know that the light-dependent reactions require sunlight and water, and they produce ATP, NADPH, and O2. Whereas the light-independent reactions, remember, Calvin cycle takes the ATP and the NADPH from the light reactions, as well as CO2 from the atmosphere, to produce glucose, right? So know the inputs and the outputs of photosynthesis and the Calvin cycle. And again, I know this is complex material. Please watch my complete review of photosynthesis so that you can master this subject. But what I want to show you is that photosynthesis occurs in algal cells, right? Like algae, because algae have chloroplasts. But Did you know that photosynthesis also occurs in bacteria? Bacteria do not have chloroplasts, right? But they do have these pancake stacks called thylakoid. They do have granum, or I should say, these pancake stacks called granum, made up of stacks of thylakoids. And those thylakoid stacks are where the light dependent reactions of photosynthesis occur. And those depend on these molecules called chlorophyll molecules. Chlorophyll molecules are what capture the sunlight in order to drive photosynthesis forward, in order to allow the light dependent reactions to occur. The Calvin cycle occurs in the stroma, the fluid outside of those pancake stacks. So if this is a chloroplast, the pancake stacks are where the light reactions occur, and the stroma, the fluid outside, is where the Calvin cycle occurs. So that's what I really want you to know about photosynthesis for microbiology course. I want you to know that photosynthesis occurs in the chloroplasts of eukaryotes, including algae. However, it occurs in the thylakoids. It occurs pretty much in the membrane, the cell membrane and the thylakoids of prokaryotic cells. Because remember, cyanobacteria, which are photosynthetic bacteria, they don't have chloroplasts. The other thing I'd like you to remember about photosynthesis for my reasons, for my class, is that, or what are the inputs and the outputs of the light reactions, the light dependent reactions, and the light independent reactions, or Calvin cycle. So do you remember the light dependent reactions require what? They require water and light. And what are the three products? ATP, NADPH, and what was the third one? Can you beat Wicket? That's right, Wicket. Oxygen for us to breathe, of course, as a byproduct. So know the ins and outs of the light-dependent reactions, and then also the ins and outs of the Calvin cycle. Remember, during the Calvin cycle, what is required for the Calvin cycle? How about the ATP that you made during the light reactions? How about the NADPH that you made during the light reactions? And what else is required for the Calvin cycle? What else is a reactant or something that's required for the Calvin cycle? Can you beat Wicket? That's right, as always. CO2, obviously. This is the whole purpose of carbon fixation, right? Taking CO2 out of the air, right? Taking CO2 out of the air and reducing it to glucose, right? So the product is glucose, and the reactants are CO2, ATP, and NADPH. And remember, where does the Calvin cycle occur? In the stroma, right? See? In the stroma, the fluid outside of the pancake stacks. In a eukaryotic, let me quiz you on this. This might be on the exam. In a bacteria, where do you think the Calvin cycle occurs? Remember, they don't have chloroplasts, so they shouldn't have stroma. That's right, Wicket. It's going to be in the cytoplasm. Isn't that neat? So you should know the differences between eukaryotes and prokaryotes in terms of how photosynthesis occurs, because this is a microbiology class. So we need to focus on prokaryotes as well as eukaryotes. And with that, our final concept check. Let's see if we learned something. The main job of the chloroplast pigments during photosynthesis is to capture photons of light. Yes, right, that's what they do. They allow the light reactions occur because they are excited. Their electrons go to a higher energy state when photons of light strike them. So it's not B because they don't absorb carbon dioxide. It's not C because they don't fix carbon dioxide into large organic molecules. And it's not D because they don't produce ATP. So it has to be A. And with that, we end this very complex chapter. And again, I'm sorry that it's complex, but I gave you 3 awesome videos that you can watch as strong background so you can really build your foundation of knowledge. If you do go back and watch those videos, I really do break down enzymes. I break down cellular respiration, fermentation, all that good stuff, and photosynthesis. So if you really want to understand this material, you can. Otherwise, hopefully you already have a strong background, but I do appreciate that this is a difficult chapter. So don't, beat yourself up if it was a little bit confusing. It take some time and effort to really flesh it out. Also, when I do the exam review video for my class, I'll make sure to highlight and stress the important parts to understand or remember for the exam. So I'll be able to help you out in that way. Other than that, thank you for joining me, and I will catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.